Parkinson's disease or PD is the second most common neurodegenerative disease, affecting 1.9% of people aged 80 and above, and 10 million individuals worldwide. PD is associated with movement disorders, caused by the loss of dopamine neurons, which are neurotransmitters required for movement. A lack of dopamine results in symptoms of PD such as uncontrollable tremors, slow movement, and stiffness. Pathogenic LRRK2 mutations are the most common genetic cause of PD. People who have Asian-specific variants of this mutation have a 1.5 to 2-fold increased risk of getting PD. LRRK2 triggers toxic changes in brain cells that are responsible for producing dopamine. However, how do LRRK2 pathogenic mutations affect PD pathogenesis is not fully understood. We previously identified the amyloid precursor protein intracellular domain as a LRRK2 substrate that contributes to LRRK2-mediated neuronal cell loss in PD. APP is one of the pathogenic factors of Alzheimer's disease, which is the most common neurodegenerative disease, affecting more than 10% of the elderly aged 65 and above. AD is characterized by the extracellular amyloid plaques, produced by APP. AICD is released into the cytosol following beta and gamma secretase cleavage. In our recent study, we have further identified a feed-forward loop between LRRK2 and APP. Firstly, mutant LRRK2 phosphorylates APP and releases AICD to the nucleus. The nuclear AICD works together with the FOXO3A protein to induce transcriptional expression of LRRK2. LRRK2 in turn phosphorylates APP, thus forming a self-sustaining loop. The LRRK2 accumulation induced by this cycle further activates a series of vicious cellular responses, including phosphorylation of RAB10 protein, excessive mitophagy activity, impaired mitochondrial, increased alpha-synuclein, and decreased tyrosine hydroxylase. All the above events result in dopamine neuronal loss and eventually neurodegeneration. Remarkably, we have discovered a small molecule drug, etanaprosid, can break this vicious cycle and restore dopamine neuronal loss. Etanaprosid is the only known AICD inhibitor, which shows beneficial effects for mild cognitive impairment. Etanaprosid binds to AICD and blocks its nuclear translocation, thus stopping the feed-forward cycle. These findings suggest that etanaprosid could have the potential for the treatment of both AD and PD. The LRRK2 AICD self-sustaining loop is highly interesting because it connects AD and PD on a common pathway. This important discovery opens the way to finding a common therapeutic target for both AD and PD, which are the two most common neurodegenerative diseases globally. This novel drug has the potential to improve and preserve the function and quality of life of those afflicted with these diseases.